I am going to talk about plant biology. I was trained as a mathematician and used to work on primates and yeast. 8 years ago, I could not imagine I would be working on plants, but I got interested in some big issues. As we know, for various problems, that factors progress in plant biology is much slower than biomedical sciences. And so there are many fundamental problems remain to be solved. And a great challenge of the 21st century is food shortage. When we eat so much, we probably don't realize that, but it's a big issue. Okay, so there's an international project, the C4 rice project. And because C4 photosynthesis is more efficient than C3, and C4 plants are more heat and drought tolerant, and C4 plants use less water and fertilizer. Unfortunately, many major crops like rice, wheat, and soybean are C3 crops. So the idea is that if we can engineer these crops into C4 crops, then we can increase our productivity. And this might lead to second green revolution. Okay. And the major reason why C4 plants are more efficient than C3 is because of the Kranz anatomy. That's a German word, okay? And so I explain what is a Kranz anatomy. Okay. So regardless of C3 or C4, you all have the vein, okay? And surrounding the vein is the bundle sheath cells. That's important, okay? And then beyond that, that's mesophyll cells, okay? That's the same for both C3 and C4. But if you look more carefully, you'll see that in the case of mesophyll, it's chaotic, but in the case of C4, it's well organized, okay? So one very regular there. And another difference is that you see that the bundle sheath cells in C3 plants don't have chloroplast. They cannot participate in photosynthesis. But in C4 plants, the bundle sheath cells have chloroplast. In fact, it's a more important cells for photosynthesis. And because of this, that it can concentrate CO2 pressure higher, and so it can reduce the oxygenation of carbon better, so it's more effective. Okay, and so, but the big question is, what are the key regulators of the development of Kranz anatomy? Okay, and so that's my major goal, is to identify the key regulators of Kranz anatomy. Okay, and we start with anatomical and transcriptional dynamics study in maize. That's our C4 model organism. And I will emphasize the important or the potential using time course transcriptome, and you'll see later, okay? So we start use maize, and so during the germination, we start with a seed, it's dry seed. So that's important because I want to use that as a reference, okay? And then you put in water, and then we take the transcriptome every 6 hours until 72 hours, okay? And here is anatomy. So in dry seed, you already have some rudimentary, the first leaf already have some rudimentary Kranz anatomy. But here you can see after 12 hours, you can see some Kranz anatomy. So here is a vein, and surrounding that, that's a bundle cyst cells. Okay. So the conclusion is that when develops a bundle cyst develop very rapidly in C4 plants, and these are the 13 transcriptomes, and 3 or 4 years ago, that's a substantial number of reads in each transcriptome. Okay. And these 13 transcriptomes can reveal transcriptional dynamics of genes and functional transition during leaf development. Okay. And here we do know a little bit about what control the cranial anatomy development. So here is a gene called a transcription factor called a shootroot, okay, SHR, okay. And so this is something we know, okay. And so we can look at the transcriptome to see how fast the transcription of this SHR gene increase. And you can see at 30 hours, it already upregulate very high. So if we want to look for the upstream regulator of cranial anatomy, we have to look genes transcription factor that's developed earlier than that. Okay. And the first thing we also did one thing, we predict the transcription factor and binding site interaction. I was very surprised that in up to 2014, there were only about 20 transcription factor binding sites known in maize. Okay. Although maize is an important crop and genetics is fairly well known. Okay. And so we expand the 13 transcriptome to 22 transcriptome because we want to have identification of close co-express the gene sets. Okay. And so these are from dry seed to our 192 hours, 8 days. Okay. And we developed a new method to predict TF-BS, the transcription factor binding site. Okay. The method, we use some traditional idea, but we add one thing, is that we add that the gene set should have the same goal term, okay? So that we want to make sure that the genes in the gene set are involved in the same function, okay? And so that's actually, that makes our method significantly better than traditional method, okay? And so first we, like other people did, you look for overrepresented motif in the promoter region of the gene set, and then you consider evolutionary conservation, and we consider sorghum, millet, and rice, and brachypodium, and as the reference species for determining conservation. And for example, this goal function, DNA-dependent DNA replication initiation, we found an overrepresent motif which is conserved over species. And so so that's one example of, so in total, we identify 1,340 TF-BS. But for this to be useful, you need to know the coordinate. For a transcription factor binding site to be useful, you need to know the coordinate TF. And we developed two methods to predict the coordinate TF of TF-BS. Okay, they are fairly cannot be explained in a short time, but in total, we predict 253 non-redundant maize TF-BS pairs. Okay, so that significantly increased our knowledge of the regular system of maize. Okay, and leaf development. And we select 12 families to test our prediction. And here I show you 4 cases. All 12 predictions validate our predictions. And now we want to infer the upstream regulator of current anatomy development. And as I mentioned to you earlier, that SHR is one known factor, okay? So we want to take this, and also here is the vascular development, and we know auxin is a most important plant hormone. It initiates gene division, cell division, and so this is auxin ARF is auxin response factor, and it's MP. And here HB8 is also important. It's a homeodomain gene, okay, homeobox gene. And here we take the approach because current anatomy develops also under total darkness. It's developed under the normal day-night cycle, but it also develops under total darkness. In fact, it develops faster because they want to struggle to see if we can receive light, okay? And here is one example how one may look for light-independent gene regulator network. So here is one gene, and in a day-night, 2 genes. In a day-night cycle, the correlation of their expression pattern is 90%. And under total darkness, it's also 90%, 92%, okay? So for this kind of case, we say their co-expression is independent of light effect, okay? And we take the minima correlation of 0.8 in both situations. We select such genes to form the co-expression network, okay? So here, the network is huge, and here we present only the transcription factors. And each dot is a transcription factor, and the size of the dot is the number, the connectivity, okay? And each line is a interaction between, co-expression between 2 genes, okay? And this one, so we have a transition here. Let me explain to you. Okay, so we take 2 genes that are most distant. Their expression are most different. And so for example, these are the 2 genes that the expression are most different. Okay, and because we have it's a time course transcriptome, so we know which genes is upregulate earlier. And so, for example, here is this upregulate earlier, this blue line, green line upregulate earlier. So we start from there, and this should be the last, okay? So then we start this, and then we say at each label, what are the genes that are upregulate? So then, I mean, you can have a network, and you can stratify, put into different labels. And there's a breadth first search algorithm. You can do that. Okay. So we have time course. And so, as I mentioned to you, we want to look the upstream gene of SHR and upstream gene of HB8 and upstream gene of MP5, okay? I mean MP1, okay? And so we look backward, and so by that, okay, so by that we have a co-expression network upstream of the 3 genes, okay? And have this, the connection are based on correlation, and we know correlation doesn't imply co-regulation, doesn't imply none doesn't. So that's why the transcription factor and their coconut, that a gene and its transcription factor, coconut factor is important. So we try to connect. So for example, here we can see that this gene has a binding site of this gene. That increase of this transcription factor, that increase the reliability of the connection. Okay. So by that, we can, this network, we can distinguish, like say for this one, we don't know its binding site. But for this one, we know it's a binding site, and here the promoter is a binding site, so this correlation seems reliable. So by that, we can see where further work is needed. So like, for example, we will be try to infer the binding site of this particular binding site and then see if they are present in the promoter. And we can make some functional validation. Okay. So the next step is, of course, we have a prediction, we have predictions, and how do we validate, okay? And so the one way is that you do loss of function experiment. So it's a mass is a C4 or minute is a C4. We can see if we knock down this gene, this transcription factor, does it have the expected effect, okay? And then in rice, we can test gain of function. Okay, rice is a C3. It doesn't have the cleanse anatomy, and if we transform the gene into the rice genome, what kind of effect it has? Does it help to form cleanse anatomy? And here are the various methods we are trying to do, okay? And here, so for example, we have this MIP 117, and we predict its control 7 very important transcription factors according to our prediction. So we make EMSA, okay, to see if the prediction holds, and 5 of them we can verify, except 2, okay. We don't know why, but we are still trying. So by this kind of effort, we can establish the connection between a different layer of transcription factor, okay. And another thing we are doing is, as I explained to you, the bundle seed cell and mesophyll cell are very important in terms of photosynthesis. And so we want to know how bundle seed cells are developed, okay, the initiation, okay. So we want to know the origin of BS cells, okay, whether it's from ground meristem or it's from procambrium, okay. And this is a machine we can use to do laser guided capture microdissection, okay. And here actually the early embryonic cell, the LCM is very difficult. Even the leading expert say they could not do that, okay? And my laboratory postdoc spent more than half a year to establish this technique, okay? It's not a single cell. You can circle to a small number of cells, and then you circle it, and then you can take out, okay? So this one example that we can take out the cell, okay? And then one big issue is band density, okay? So this is, you can see that the structure of C3 is different from C4, okay? C4, you have a very small space in. C3, you have a 7 mesophyll cell in between. C4, you have only 2 mesophyll in between 2 bands, okay? And so the question is, what gene control this development, okay? And here show you an example. So this is a C3, and in the development, this means the order of band development. This third order, fourth order, and it stops at sixth order. And why C4 band density is higher is it goes up to seventh order. So our purpose is to see how this difference occurs. And it's known that oxygen maxima, if oxygen accumulates high enough concentration, then it initiates the band development, okay? And here is how oxygen are synthesized. There are 2 ways, okay? And this is a tryptophan-dependent oxygen synthesis, okay? And here, what I want to show is that before, so this color bar, okay? Red is high in C4 plant. Blue is high in C3 plant. And before this charismata, you can see that there's no differentiation between C3 and C4 plant. But after that, then you can see that it's a red. So its expression is higher. And so that may explain why oxygen level is higher in C4 plant than in C3 plant. Okay. So our idea is, okay, what are the genes that control the synthesis of oxygen? Okay, for example, this UCA, YUC, okay. So if we raise the expression level of UCA in rice or in a C3 plant, can it increase the band density? Okay, that's our idea, okay. And so actually this is the C4 rice project is a 15 years project, and there are still many fundamental problems to be solved. Okay, and this we have a computational genomics team and a molecular biology team, and because of time limitation, actually some other team, but I'll stop here. Thank you for your attention.